Now, what we're also doing, because we're interested in producing libraries, many of the people we wish to work with want to actually knock out entire panels of gene families. We're also turning around and creating limb systems. And again, we're looking at open source in order to be able to come up with various solutions for being able to design limb systems. So in this particular case, we're looking at the ontology community. So the ontology community is basically allowing us to look at, say, how does one design an assay and describe an assay? We're looking at organizing our workflow elements into nodes and edges. The nodes consist of things like the actual experimental items we're going to produce, oligos, plates of oligos, pooled oligos, clones which are derived from the pooled oligos. We're also defining the edges between those interactions. How do those actually come together? Okay, what are the things which have to precede an experimental event? What is succeeded by that experimental event once it actually is passed and validated? And then finally, how do we actually track all of this? Now, this part of the software is actually commercial. This is something called Nautilus Limbs. But many of the ideas that come from Nautilus Limbs actually are in the open source community. Okay, and in many ways, whenever we were developing this workflow ourselves, as we were going through these individual steps, these are individual experimental steps which we're replicating in the software. What we're actually doing here is also tracking all of that and using these descriptions of how to do an assay or how to do an experiment, bringing that into our software and making sure that what we're generating is both, one, experimentally valid, and two, is actually to be computed across. Okay, so we can be sure the results coming out of this are actually consistent with what we actually wanted to get in the first place. Third part of this is actually detect. Yet again, more open source software is starting to be involved here. So what will end up happening now is that this is a very simple example. You have a control. You've amplified your control. You now hit your control with your guide RNA and your Cas9. What's happening is your single band is now being cut. Okay. And what we're using are softwares, a variety of different softwares. Some of them are commercial, belonging to the instrument, which is the gel reader. Sometimes we're using things like GIMP in order to be able to detect what is the ratio between this band compared to these two bands. The ratio of these bands then gives us a percentage likelihood that cutting has actually taken place. Okay, and these are all then, again, validated with the various different systems we have for being able to do the experiment in the first place. And if you're interested in it, there's a paper that came out last year from our laboratory, Sequan Liang. It's in Journal of Biotechnology, and you can follow up on sort of like many more of the details of how we actually do this. Here you can actually see it's not enough to just do it with one cell line. All cell lines are different, okay? And what is different about cell lines is that sometimes the nuclear availability of a particular site that you want to cut might actually be different depending upon the cell line. Some cell lines grow really easily and transform really easily. Some cell lines actually grow poorly and are really hard to transform, okay? So what we've done is basically tested these against a very wide range of different kinds of cell types. And what I'm going to draw your attention to is two things. Basically, those are the different cell types, and we're using a couple of different types of transfection systems in order to get our technologies, our CRISPRs, into the cell lines. And what you can see again as you're going along here is that some technologies and some cell lines are really, really hard to cut if you only look at one particular application, if you only look at one way of doing the experiment. But what we're finding is that, again, being able to combine both of our experiments and being able to combine a much wider range of reagents, what we can do is we can actually make a recommendation to you as to what is the best reagent to likely use for your experiment. Okay, and what's important about that is basically it means that whenever you come to us and you ask to use something or you want to use something, we can actually have an intelligent discussion with you now. Okay, and basically tell you what we have found based on our research, and also be able to tell you what is most likely to work well within your hands. And indeed, if you find that this is not something that you want to do by yourself, we also have services. So what our own scientists can actually do is look at our own internal database of what works and what doesn't work, and again have a much higher probability of being able to get to a successful outcome of an engineered cell line faster than we ever did before. This is an example of coming in and using these technologies not to do single knockouts, but actually to multiplex knockouts. Okay, And again, this used to be a very, very difficult thing to do. It was very fraught with difficulty and fraught with actually it not working properly. Here, however, what we're able to do is we're reliably able to go in to multiple loci, knock them out at the same time. And we're able to see that the way in which they're being knocked out, the efficiency with which they're being knocked out is actually really quite similar. Okay, you're looking at between a 60% and a 90% cutout for each, or knockout for each one of these particular individual genes, which was actually hit by this technology. And again, remember, this is all done in the same test tube at the same time. Then finally, if you want to go in and actually profile what's going on, okay, again, what a lot of the stuff in our laboratory is now involving is basically going in and precisely characterizing the change that you made to that genome. So in this particular example, we have CE sequencing. Going in, amplifying the region that you hit, okay, pulling out the PCR product, sequencing that directly, and being able to go in and see exactly where the indel was made within the genome. And in some cases, that's actually an important part of somebody's workflow. You can also do this in NGS. So again, the thing to emphasize is we're using a lot of different softwares out there. We're using SBAL as a data model around which we are building our tools and reagents. And we're basically standardizing the way in which we do our experiments, okay, and as much as possible to make it as efficient and easy for somebody to do these kinds of experiments as possible. And the result of it basically is we're able to generate a 4-day workflow for engineering an individual cell line. Now, looking at these individual delivery formats, again, to emphasize the point, we've got 4 different formats. Now, if I was a biologist in the laboratory, me sitting and look at this, I would have to work with somebody in order to figure out what the best way of doing this is and why, okay? An awful lot of the time, this is actually anecdotal evidence you only learn by doing experiments in the laboratory with somebody. You may find out, after working with us for a while, that in fact the best approaches are either of these two, and you then may be guided towards particular ways in which you should do that experiment. Okay, now, the cool part about this is because we've actually turned around and we spent so much time characterizing and validating the workflows for knockouts, it's actually made the process of doing knock-ins much easier. Okay, so because we've worked out the rules of how a knockout is done and then how to join 2 pieces of DNA together using homologous end repair, what we're doing in these 2 experiments, we've got the same cell line, okay, and what we're doing in both cases is we're introducing a new type of fluorescent protein, and we're looking for the presence of that fluorescent protein within the cell line. And indeed, we're looking for a green fluorescent protein in this experiment. We're looking for a blue fluorescent protein in this experiment, and in fact, we have a combination of both in this 3rd cell line. So again, what we're now starting to do is standardize the ways in which homologous end repair is performed and homologous end joining should be done so that you can actually have a higher likelihood of getting a successful experiment by adding a new gene, okay, using exactly these technologies. And what's that going to ultimately involve? Actually, what it's going to involve is basically understanding the model of how insertions should take place once you do a cleavage with a Cas9 or a talon. It's going to involve looking at what are the best places to introduce a donor and what are the best donor designs to actually use. And it's going to result in models for how you actually do this design, okay, which will then again be built into our software, okay, and again built into our validated reagent sets so that you can actually do these kinds of experiments in the most efficient way possible. So, conclusion really is there are an awful lot of benefits to us which have come to us through using synthetic biology practices and approaches, and also by using the way in which software has been developed to support synthetic biology over the last years. Okay, there are an awful lot of benefits which allowed us to streamline our gene editing program in order to maximize how can we design good guide RNAs, in order to maximize how you actually do the protocols to actually make that, emphasizing the protocol has to be standardized, has to work reproducibly. Ideally, there should be data around the cell lines that you're actually hitting with that particular protocol and how you should do that protocol for an individual cell line type. And what basically the key to determine what is the best likely combination of technologies that will allow you to do your experiment. So again, think about it. You go and you look at the methods paper of any given paper which is published today, and you have a variety of different tools, reagents, and sources from a whole bunch of different places. Now again, one of the advantages of ThermoFisher is because we're so large, we actually don't have to leave our own company anymore. And we actually think that that ability to produce and optimize how to do an experiment and then deliver that to end users is actually a big benefit. Now, the software side of this, and again, this gives you a sense of sort of like the various different tools, reagents, instruments, whatnot that we actually have available all under our own umbrella. Now, going back to our guide RNA questions, I'm going to leave you with one set of ideas. So, when you're looking at a guide RNA, you're getting a number of different pieces of information. You're getting the sequence, which will actually be bound to your genome. You're getting the information about how many targets that particular PAM might, or that particular guide RNA may actually have, so-called on targets and off targets. You're getting information about the location of these various different genomic loci. Okay. And you're basically getting a sense of sort of like where exactly might this sit down, where might it also sit down in an off-target way. And you're getting a series of different primers which allow you to specifically test. Okay, so again, these are being designed on the fly based on the genomic sequence which is available to you. Now, going through here, basically these are more of our assays which we're getting set up here for the customer. All of this is done in an automated way. So it used to be involving people going to 3 or 4 different parts of our website. It's all done in one tool now. Now, the advantage of this is starting to be that if you look at synthetic biology open language, particularly the data model associated with it, there's a couple of very nice synergies which are starting to take place. So SBAL basically supports information about the sequence and the sequence design, okay, the thing you're actually making to put into the cell. But SBAL also contains information about what it's going to do once you actually get it into the cell. So we have a very nice synergy where we have sequence information, and this basically overlaps the traditional molecular biology crowd. Okay, and again, I have a software, Vector NTI Express Designer, which is a synthetic biology software. But nonetheless, we have the ability to bridge not just the traditional molecular biology, but also potentially taking this information about our guide RNA and its on target and potentially off targets and pushing this information into softwares like, say, Cytoscape, which are designed for pathway analysis or pathway design. Okay, so it's a potential bridge format. And what, lost that slide, never mind. What I would suggest actually is a potential powerful outcome for synthetic biology, particularly within the cell engineering workflows, is not just the ability to say these are potentially pathways which are going to be disrupted and being able to simulate what the phenotype is going to be in my cell before I do my experiment and then compare the phenotype I got versus the phenotype I expected after I did my experiment. But SBAL potentially with some extensions could also be a very nice way of being able to define all the constructs I put together. So if you think about it, each one of the constructs we made within our LIMS system, there are 20,000 genes targeted. There are 4 targets per gene. That's 80,000 individual constructs that we have to track within our LIMS system. And there's many more than that when you're actually making those final list. Okay. What if there was a nice way within SBAL of being able to track the assembly of things? Okay, so what that would mean is sort of like as I'm making something, I can actually track how I actually made it or how my collaborator made it. Another thing potentially that would be very nice for SBAL or open source software in general is wouldn't it be nice if there was a way of actually tracking how the experiment is done? So again, we have a very simple way of doing experiments. We use all of our own reagents and we validate and verify to make sure that they all work together, and then we make recommendations on what you should use. What if you basically had a way of also doing this with your own protocols or materials, and you had a way of being able to compare this protocol versus that protocol, particularly when you're starting to go in and engineer cells or doing some kind of a complex workflow, and be able to have the advantage to be able to carry within a data model perhaps pointing towards another standard for assays or another standard for long lists of assemblies in standardized backgrounds. What if you had a way of being able to build these models? Okay, now this is the kind of thing which won't happen easily in industry. Okay, quite frankly, it's not our bailiwick, and it's also not something that we're going to spend a lot of time doing simply because it doesn't help us to move or shift reagents easily. But what we could do is work with open source communities in order to develop something like this. That's the reason why I've been involved in the SBOL community since about 2010. And that's the reason why I agreed to be an editor, and that's why I'm actually on the advisory board now. Okay, so the thing I would leave you with, where we find open source software and approaches like Nona particularly useful, is basically it allows us a way of collaborating with somebody who we can talk to have a sensible conversation about, okay, how does it work? Where can I find it? If it doesn't exist, how could we maybe go about doing this, and how might we drum up interest to get this developed? So we can at least prototype and figure out, will this actually work? And if it really does look like a useful solution or tool, then we start to incorporate it in our own tools and practices as well. So, thank you very much. And I will take any questions you may actually have at this point. Thank you.